I hit start record now. So the difference between these two was where the receptor is located. And if I kind of compare these two, I'm going to do a quick recap because I forgot to hit record. So if I compare that signal molecule with that signal molecule, this signal molecule is nonpolar. Nonpolar things have an easier time slipping through the membrane. Okay, so now what we do is we'll look at one last thing, and then like I said, I'll write some stuff on the board to kind of organize all this stuff. If I have an enzyme, what was the name of the thing that fits an enzyme? Yeah, a substrate. Cool. What's the name of the thing it's not substrate? What's the name of the thing that fits a receptor? It's not signal molecule either. It starts with an L. It's possible you haven't ever heard it. I have no idea. Did somebody say ligand, maybe? I heard something. So ligand or ligand. Down here, if you go to college somewhere kind of more in states around us, they usually call it ligand. I've heard it ligand. I don't know if that's like Northeasterners or what. So enzymes and substrates go together. Receptors and ligands go together. There's a difference. An enzyme will change a substrate. So an enzyme is always speeding up a chemical reaction. So if my hand's an enzyme, and it fits expopen, my hand's probably called expopenase, bless you. And what will happen is, bless you. And what will happen is I'll grab this substrate, and then as part of a chemical reaction, maybe I'll like break off that part or something. But the substrate goes in, and the product that comes out looks different because that was like a chemical reaction. So I'm either like sending in a couple of substrates and I like shove them together, or maybe I send in one substrate and I break them apart. Like what goes in and what comes out looks different because an enzyme is running a chemical reaction. With receptors and ligands, that's not necessarily the case. The ligand doesn't have to get destroyed. So receptors and ligands are actually a better analogy for a lock and a key, because if I use a lock and a key, can I reuse the lock and reuse the key again tomorrow? Sure, I don't destroy it. An enzyme and a substrate, as soon as I put the substrate in there, I'd like break it apart or something like that. Okay, so we sort of break these down into like two categories. One of them is polar stuff. I'm going to give you the title of them in just a second. The other one is nonpolar. All right, cool. So when I'm looking at ligands, I can generally break them down into do they behave like nonpolar ligands or polar ligands? All right, so if I'm looking up here, let me see if I got a good picture of cholesterol. There's cholesterol. So cholesterol, do you think cholesterol is polar or nonpolar? Nonpolar. And the logic of how I might know that if I haven't heard it somewhere is like, hey, I know that cholesterol can clog my arteries. My arteries are mostly water. So inside, my blood is mostly water. So cholesterol must not dissolve in water very well. Otherwise, it probably wouldn't clog my arteries. So just like saturated fats can clog the drain or clog my arteries, cholesterol must be kind of solid. So it's like waxy, and it can clog my arteries. Okay, so if I have the word cholesterol and I compare it to these words, it's possible I've shown you this before. I show it to you guys a few times throughout the year. So I'm grabbing a image really quick. When I look at the word cholesterol and then I look at the names of a whole bunch of hormones, I'll kind of notice something. Not 100% of the time, but most of the time. So how can I tell that these are cholesterol-based? Yeah, the shape. Cholesterol, if we go back and look at our cholesterol molecule, it'll look like this. It's like hexagon, 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 pentagon, in sort of like a stair-step motion. And then if I look at these, don't they have like similar shapes? Yeah, so those are cholesterol-based. Thank you. So those are all cholesterol-based. Now, ignoring the first one at the top left, what do you notice about the names? Aldosterone, testosterone, estradiol, progesterone. I'm going to write the word cholesterol based up here. Cholesterol based. What do you notice about, like, you might have never heard of aldosterone. Is there anything in the name that makes me think it might be cholesterol based? Yeah. Yeah, it seems like I keep seeing like estro, estro. Cortisol doesn't really work, but a lot of cholesterol-based hormones have like ostro or estro or something like that in the name. They're cholesterol-based, so they're nonpolar, and so we call all of these steroid hormones, cholesterol. Any sort of hormone that's made from cholesterol is called a steroid hormone. So steroid hormones work one way because they're nonpolar. This other type of hormone that's often polar is protein-based or amino acid-based, the building blocks of proteins. And they are polar. So I have two major categories. I have protein-based, and then I have steroid hormones. The protein ones are polar. The steroid ones are nonpolar. Steroids are called steroids because they come from cholesterol. And even if I saw a word that I've never seen before, like aldosterone, I kind of see the word cholesterol, stero, kind of in the word. So if I gave you a brand new chemical that you've never heard of, it's called blah, 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 ostro, blah, 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 you could be like, oh, that's cholesterol-based. So it's nonpolar, and even though you had never heard of the chemical, you could describe how it works. That's how it works. I'll emphasize like what's happening here too, but yeah. Huh? To the which one? Yeah. So if I'm looking at my cholesterol hormones, they look like that. So they kind of have like a hexagon, 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 pentagon, kind of like that. Now, you'll never have to like draw these on a blank sheet of paper, but if I showed you that, you'd be like, hey, if I just showed you a picture of this, you'd be like, hey, that's either cholesterol or a cholesterol-based hormone, so it's a steroid hormone. All right, I can recognize steroid names because they often have estro in them. How would I recognize a protein? If it was a brand new word I've never seen before, how would I know it's probably a protein? By reading it. Huh? Yeah, if it ends in IN or EN. If it ends in ACE, it's probably an enzyme, so it's not a ligand. But if it ends in IN or EN, then it's probably a protein. So it's probably polar, so it probably works this other way. It's going to work this way. So even if I've like never seen the words before, I can possibly describe about how they work. So like insulin. Do I think insulin is a steroid hormone or a protein-based hormone? Protein, because it ends in IN. So how does insulin work? Like this. Cholesterol-based hormones, steroids, are a little bit different. They work a little bit more like this. So let me kind of show you like, okay, what things matter about what's different in this diagram. So first of all, they ask this on the AP Bio test all the time. Just to kind of show you, I'll pull up a question. It's like shocking how similar the diagram is to our diagram, but this is an old free response. So I'm supposed to describe what's happening in step one, like name it and describe it. Step two, name it and describe it, all of that kind of stuff. And then if you kind of compare that to like the diagram that we're using up here from a textbook, it looks like they pretty much just ripped off that picture. So here's what matters. I kind of break it down into 4 steps instead of 3. You can call it these 3 if you want. That's fine. I always think of it as I have to send out a ligand. So step 1 for me is some cell has to send out a ligand. It has to send out a signal. So maybe this ligand is like growth hormone. And if it hits a receptor in my skeletal system, my bones, this will eventually say grow. Let's say that the signal molecule is insulin. It's going to say send a bunch of messages that will result in me pulling sugar out of my blood and feeding my cells. So step one is like send out a message, a ligand. For reception, if I'm trying to earn points, pretend I'm writing an answer on a free response at the end of the year on the AP Bio test. What should I say about reception that would get me points? I don't just say like reception happens. Obviously, that's not enough to get anything. Like, what's important to say about reception and receptors, do you think? Do what? That's true. It receives a signal. What's wrong about if I say a receptor receives a signal? What am I kind of missing? I'm not really wrong, but I'm kind of missing something important. Yeah. Yeah. So I want to emphasize that they are specific. So this is like growth hormone receptor, so it only fits growth hormone. Or this is an insulin receptor, so it only fits insulin. That's kind of like saying a substrate. You know, like, substrates fit enzymes. And you're like, yeah, but like that enzyme fits that substrate. That enzyme fits that substrate. If I just say something like locks and keys go together, I'm missing out on like that lock goes to that key and that other lock goes to that other key. So I want to emphasize that if I'm talking about reception, that a receptor is for like one specific ligand. They are specific. They have to like physically and chemically fit. Yeah. So if I'm talking about receptors, if I'm polar versus nonpolar, the thing that I would say about the receptors, they still have to physically fit. They still have to chemically fit. But where is this receptor located? It's located inside the cell because that guy is nonpolar. So this guy is nonpolar, so it's going to hit a receptor inside the cell. And we will see something cool soon, so hang with me. Receptor is inside the cell. If it's a protein-based ligand, then the receptor is in the membrane. They'll call it membrane-bound, but you can just say like in the membrane, or it's stuck in the membrane. So up here, this guy's polar, so his receptor is like stuck in the membrane. But as far as like, hey, receptor is specific to a ligand based off of shape and everything, that part's the same. Does that kind of answer your question? Yeah, cool. All right, great. So now, here's another part. The protein ones pretty much always do this. I don't know of any exceptions. They have what they call transduction. How would you describe transduction? Each one of those green proteins is different. So if you're trying to put into words what transduction is, thank you, what would you say? I'm trying to describe transduction. And you can talk with somebody around you if you want. What would I say transduction is? All right, so how could I describe transduction? I was trying to summarize it without having to write like 5 sentences or something. Yeah. Okay, passing through. If I say passing through, I agree. But if I say passing through, it might seem like just one protein is moving and passing through. But it's a protein activating a protein that eventually does something. So I think of it more like a relay race. They'll call it a cascade in the mountains. If you have like a cascading waterfall, it like crashes, falls. It's kind of like. So you want to have, oh, maybe they didn't want to call me. No, somehow the tone changed on my phone and it goes from really quiet to like slowly gets loud enough, like an alarm. and it doesn't sound like a phone to me. All right, maybe they dialed the wrong number and realized it right away. So transduction is like a protein activating another protein to cause an effect. So now the cell is going to do something. If I'm writing for the AP Bio test, I would like give an example of a response. Like if we're talking about insulin, I bring in blood sugar. Or if we're talking about growth hormone, then like the cell's going to grow, something like that. So transduction or a transduction cascade, or it's like a relay race, something like that. Over here, it's a little bit weird. Textbooks contradict themselves a little bit. So I still go ahead and say that transduction happens, but now it's just like one protein usually like moving into the nucleus to like turn a gene on or off. So now I don't have that relay race. It's really just kind of like one guy running the race. Over here, it's more like I run around the track once, a quarter mile, I hand off the baton to the next guy, he runs a quarter mile, I hand it off to the next guy, he runs a quarter mile, like that. Okay, so one last question, and then we'll be able to see like why this is useful and why it's not sucky. Can anybody think of a good reason? This is a very hard question. Why we would tell a protein to tell a protein to tell a protein to do something. That seems risky. For example, if I wanted to get a message to Miss Skull, she's way down there in South Hall. Wouldn't it be more likely to get there if I just like walked down there and told her myself than if I told Asilius, who told Coach Martin next door, who told Sterling next door, who told like all the way down? If I had to make it through like 20 teachers before it got to Skull, isn't the message more likely to get lost or screwed up? So why would we do that? That's basically what we're doing. We're playing the telephone game. Why is that worth the risk? There must be some big benefit. Yeah.
I don't know, but once it's resting, the first protein it can't do what the other protein at the end of it can.
That's true. Maybe it's like this guy has a certain talent, and this guy does, and this guy does, almost like a moving assembly line. We're building a car. I'm good at putting on the wheels. The next person's good at putting on the tires. Somebody else puts on the door, then somebody else puts on the mirror or something like that. So that makes sense, but they aren't doing anything except passing the message along. So they aren't really changing anything. It's not like they're building something along the way. What do you think, Colton? Yeah, maybe I can pass it along faster. If instead of me like walking down the hall, if I can just hand it to a guy who hand it to a guy who hand it to a guy, maybe it's faster. Anytime, let's say there's some disaster in like my home state of Oklahoma, there's tornadoes or something, and you'll see like them unloading trucks just full of bottled water, and they just have a line of people, and like the bottled water just goes down the line of people as they unload it. So maybe it's that, and that might be true. Turns out the real reason why each one of these is a checkpoint, a quality control checkpoint. So here's what I mean. Pretend that we work at Apple, and I have no idea who runs Apple. I don't know if he like wears mock turtleneck shirts or not. Let's say that I'm CEO or president or whatever of Apple, and all of y'all are like Apple iPhone engineers, and we're getting ready to come out with the iPhone 17 in a few months. I think they released it like now. Let's say it's coming out in a few months. So y'all are my engineers. What I'll do if I'm running this, before we like go and make millions of them, I'll be like, okay, you were specializing in like on a screen. So Jackson, hey, you're working on screens. Did you make the screen? Our goal was like 10% more scratch resistant. If I ask him, did we make the screens for the new iPhone 10% more scratch resistant? We all work at iPhone. What do I want to hear? Do I want to hear yes or no? I want to hear yes. So he's like, yes. And I was like, cool. That's like this. And I'm like, all right, Caleb, you're working on the batteries. Do those still explode a lot? What am I wanting to hear? I'm wanting to hear no. Cool. So I checked that. I was like, I wanted a yes there. I wanted a no there. All right, Colton, did we add like a fifth camera or whatever on there? And he's like, yeah. And I'm like, good, because 4 is stupid. 5 makes sense. And if I go through all these yeses and nos and I get the right combinations of yeses and nos, I'll be like, cool. Find a bunch of 8-year-olds in China. We're going to make millions of these. And they're like, they're making shoes. And it's like, get them off of Nike shoes, get them on our phones. Pay them an extra $1 a month. So we do that. Dark humor, sorry. So we do that and we move on. However, what if, as we were going through there, I'm like, hey, engineer, are those batteries still exploding? And you're like, bigger than ever. It's like a bonfire. Should we proceed? Now, maybe we do. Maybe we don't. But we shouldn't. That's what this is doing. You're like, cool analogy. How does that relate to an actual cell? Pretend that this is growth hormone. Growth hormone makes you taller. So I'm 10. I'm supposed to go through a growth spurt over the next few months, and I'm going to get taller by a couple of inches. Okay, if I'm going to get taller by a couple of inches, doesn't that mean that my skeleton needs to get taller, like my bones need to get a little bit taller? Don't bones require a lot of calcium and stuff? What if I was in taking 0 calcium in my diet? Is it a good time to go through a growth spurt? No. All right. Muscles. If I'm going to get taller, if my legs are going to get longer, don't like all the muscles in my leg need to get a little bit longer also? Yeah, I need amino acids for that. What if I'm getting 0 amino acids? Is that a good time to grow? No. So the way that it works is it might be like, hey, you're 10, and my body decides growth spurt time. We send out a ligand, growth hormone. It hits that specific receptor. This protein is around if I have enough calcium. This one is around if I have enough amino acids. This one's around if I have enough of something else that I need in order to grow, maybe like lipids and fat and stuff. But if I'm missing any of those, then now I don't grow. So they're all a checkpoint. Protein 1, protein 2, protein 3, protein 4 on the iPhone. And once I get through like 32 proteins, if I get all the right yeses and nos, we make iPhones. But if somebody along the way in the mix basically is sort of representing no, then we don't. So there's another chain of proteins that are doing all this stuff to make this one if calcium is present. If calcium is present in my body, calcium will hit a receptor and it'll say, make this. If plenty of protein is present in my body, some amino acids will hit a chain of proteins and it will result in this. So if calcium's around, he's around. If protein's around, he's around. If something else I need is around, he's around. And now I only try to grow if I have all the ingredients that I need. Yeah.
So it's like a combination lock?
Yeah, kind of like a combination lock. Not only do I just spin it once, but it needs to be 10, 20 to the left, 35 to the right. Like I need 3 different numbers, or I need 10 different numbers along the way. Does that kind of make a little bit of sense? You might be stuck on like, how does the cell decide that this is on, or how does it know it has enough calcium? That's a hard question for now. Take a moment. So as the year goes on, you'll get better able to answer that. But now take a look at this one. Everything above this is outside the cell. This is my phospholipid bilayer. Everything above this is outside the cell. Everything in here is inside the cell. All right, cool. Take maybe 30 seconds. Talk with somebody if you need to. See if you can figure out what's going on. And this one will be slightly familiar because you've probably heard of insulin. So if you can't quite read that, that says insulin. So see if you can figure out overall what's going on. You don't have to say like, this protein activates this protein, activates this one, this one, this one, but just like overall, insulin hits an insulin receptor, and then like, what's the result? So talk with somebody around you. All right. So whenever insulin hits an insulin receptor, what's like the final outcome? Yeah. Do what itself? Yeah, glucose gets let into the cell. So most people that talk about insulin, they're always like, hey, insulin lowers blood sugar. That's true, but that's not the point. So when I eat a sugary meal, and now there's a bunch of sugar in my bloodstream, great. But that sugar in my bloodstream has to go into my cells, otherwise I starve and die. So if I'm diabetic, the reason that I die if I don't have insulin is my cells starve. Imagine the hallway is the bloodstream, and each classroom is like an individual cell. So there's a whole bunch of like sugar floating through the hallway. We're a cell, it's not entering, so we are going to starve and die. When insulin hits a receptor, it's got the right shape. That insulin ligand has the right shape, kind of right chemical character to like fit this receptor. Transduction happens. Protein activates a protein, activates another. I then ship some of these glut transporters, and now all of a sudden, I open up the door and some transporter starts like shoving glucose into the cell. If I'm in a hospital, we're always talking about what's this person's blood sugar like. But if it's really, really high, that means they're starving because the sugar is staying in the blood and we need it to go into the cells. So the labs that we do are measuring blood sugar because we can do that with a blood test. But what it means, if my blood sugar is super high after I eat and it keeps getting higher and higher and higher, that means the sugar is not going into my cells. I'm not diabetic. So if I eat sugar, my blood sugar spikes, I release insulin, and then I'll notice that my blood sugar goes back down. That means I'm shoving that sugar into my cells and I'm not going to starve. So insulin release causes all this to happen. What I'm trying to show you here is this is very kind of meta. You're like, all right, insulin hits the receptor and now sugar enters my cells. And you're like, but how does this protein tell this protein to like have sugar enter your cells? Like, how does it actually do that? Like this. The final step is I have these sugar transporters and they're just waiting. And if all these messages go through, I ship them to the cell and I start transporting glucose in. So this is just trying to show you an example of like, how is it that a protein activating a protein does something? This is how it works with blood sugar. Does that make a little bit of sense? So we're just a bunch of chemical reactions. Now, they're like, okay, that's a little bit complicated. That's more complicated. That's like a longer transduction cascade. Turns out you don't have to know this for a test or anything, but it turns out there's a bunch of different sort of receptor types. They all behave differently. So kind of like at school, you've got like band, and if I'm a band kid, I might behave differently from like a theater kid that might behave differently from a softball player, from a baseball player. There's kind of certain things that I expect. So here are these like common receptor pathways. Some of them have crazy names, like hedgehog, the hedgehog pathway or the notch pathway. The reason that some things have crazy names, the reason that some genes have crazy names, is if a gene is discovered in a fruit fly, the person that discovers it can name it anything that they want. If a gene is discovered in anything else, there's all these rules. So if I'm studying mice and I discover some gene that does something, I don't have much control. It's going to be called GP7349 gene. However, if I'm a fruit fly geneticist and I have fruit flies and there's some gene that gets mutated, and anytime that gene gets mutated, they have these giant spiky hairs that grow out of their back, I'm like, looks like a hedgehog. That's the name of the gene. And so there's a whole hedgehog family. There's sonic hedgehog. That's the original. I'm not making that up. SHH is the sonic hedgehog gene. Mutations in humans in the sonic hedgehog gene often cause you to have limb deformities. This is where you'll have like 8 fingers on a hand or something, or like your hand is almost like a mirror image, where it's like pinky to thumb, and then all of a sudden there's like a ring finger and a pinky kind of sticking out here. All kinds of weird abnormalities like that. Really fast, do you want to see the hedgehog mutation in a fruit fly? Give me 1 second, I got to find it really fast. Hadn't planned on showing it. It just sort of like popped up. Ooh, this one I don't use as often. Gene regulation. It's kind of cool looking. So this will be a fruit fly. So it is a tiny fly, but he's under a microscope, and so that's why he looks so big. And again, the first one that they ever discovered, they named Sonic Hedgehog. The picture I'm looking for. Sonic, so there's this Nintendo system, or a competitor of Nintendo called Sega, and they had a character named Sonic, and he was a hedgehog, so they named it after a video game character. All right, I got one more spot. Development. Oh, this is going to be the right spot. There you go. That's a fly with the mutation, SHH, Sonic Hedgehog mutation. They don't normally look like that. I realize that you haven't seen fruit flies up close probably, but normally their hairs are like, I don't know, 1/5 that long or something like that. It just looks like peach fuzz. And again, when humans have this deformity, they start growing, or chickens or other things, they start growing like double hands. It's like double hands. This is a chicken wing. So I've always thought maybe after I retire, I'll open up like a chicken wing store. And the way that I'll have a leg up on my customers is I'll make sure that all my chickens have the mutation. So I get double chicken wings out of every chicken. People will be like, we don't know how he does it. He only orders this many chickens, but he has this many chicken wings. Yeah. So kind of crazy. All right, so that's the hedgehog pathway. Notch receptors, when fruit flies have that, they have all these weird notches in their wings. One thing that's controversial, they've talked about doing away with all these names because some of the names are like cuss words and everything else. One of the names is like lush. A lush is somebody that basically is an alcoholic. And so there's this one gene that if you're a fruit fly and you have that mutation, you love alcohol. Turns out humans have that exact same gene, and they love alcohol if they have that mutation. And so sometimes this gene in fruit flies, about 75% to 80% of genes found in fruit flies are found in humans also. So you're like 75, 80% fruit fly. So they'll name some gene, like there's not one actually called butthole, but let's say they have this gene named butthole or whatever, and it might be a cause of like colon cancer or something in humans. And you go to your doctor, this is the controversy of why they're like, you shouldn't name these genes. And they're like, what's wrong? And it's like, well, your butthole gene is mutated. And they're like, I'm offended. And it's like, I didn't name it, but it was found in fruit flies, and they named it this, and that's what it is in you. There's a gene called I'm Not Dead Yet, based off of a Monty Python film. And the I'm Not Dead Yet gene in fruit flies causes their lifespan to double. But in humans, that mutation might shrink your lifespan. And so you might be at the doctor and like, what's wrong? And you're like, oh, you got a mutation in this gene called I'm Not Dead Yet. But for you, it means like you're going to die sooner. And so when you're having these conversations, it's like not as funny. And so they're like, hey, maybe we got to like slow down fruit fly geneticist, things like that. So in a cell, it gets a little bit more complicated. So we see sort of a simplified version of transduction. We see a cell with just the insulin pathway in there. But then in real cells, like you've got multiple pathways going on like all at the same time. It's like every single 2 or 3 letter abbreviation up there is some protein. So this is like how a process works. If you're like, hey, at the truly like super detailed level, like how does it work when our cells want to do this one thing? And you're like, this. Oh, what about this other thing that my cells do? Oh, well, the pathway for that one is this. Back there, I've got a poster. It shows how nerve cells talk to nerve cells in normal brain cells. Then it shows how nerve cells talk to nerve cells if I have Parkinson's syndrome. And it basically shows like this protein activates this one, this one, this one, and this one, and oh, you got Parkinson? Here's where the problem happens. It's almost like they'd be like, here's how a cell's supposed to work. And I've got disease A, and because I have disease A, that guy is not doing his job, or this guy drops the ball, something like that. So as I will say many times throughout this year, I don't understand how I'm still alive. If doing one thing requires that, and then doing another thing requires that, and doing another thing requires that, like I don't understand how I'm still here. That's so complicated. I don't understand how it doesn't screw up more often. All right, so the main thing that you need before the application part is I've got maybe a signal molecule, a ligand. It's a specific receptor. They have to fit. I might activate a guy that activates a guy that activates a guy. That's called transduction, when proteins are kind of relaying the message along. And then the last thing would be the cell responds somehow. Maybe I turn on a gene that says grow. Maybe I turn off a gene that says quit growing, or something like that. So we're just a bunch of chemical reactions. If I'm cholesterol-based, a steroid hormone, the main difference is now I hit a receptor on the inside of the cell. And now transduction probably doesn't have a whole bunch of proteins activating each other. It's just that receptor goes in there, turns some gene on or off, and then my cell responds by like doing whatever it says to do. If it says make adrenaline, then I'm now like building adrenaline or something like that. Yeah, Thor.
I think that, so protein hormones are more common than steroid hormones? Or
I
like
don't know the answer to that.
Okay, like do you think we do more transduction than we do like have other?
That is true. So because that's true, I'm guessing. But I do, anytime I'm like looking at pathways, it's almost always a polar molecule. So yeah, if I had to guess, I would say it makes a lot of sense that protein pathways are way more common than steroid pathways. Again, I've never read it, but now that I'm thinking about it, when I start looking at pathways, most of them are that kind of pathway, a polar pathway. So that totally makes sense. I'd have to Google it before I said I'm 100% sure, but like that totally makes sense. I think that's right. Any other questions so far? All right, now, we actually just finished the boring part of this unit. The cell membrane stuff is the less interesting stuff. Organelles are way more interesting, so now we start the organelles part. When we start organelles, I start a whole new cell diagram. So if you're the type of person that kind of draws when I draw, you'll want like a new one. You might be tempted to try to shove the organelle stuff in the middle of your old diagram, but you probably won't have room. So I would go ahead and like start a fresh big old cell diagram. All right. Here, I will be teaching all the organelles with basically just one giant diagram again. However, this time, following along in your notes isn't like as hard if you're like, I want to follow along in my notes and I want to look up at there at the picture. It's much easier than with cell membrane. So as far as where you'd go if you're wanting to follow along, that's page 1. Page 2, I'll move over here to the camera. Page 2, that's page 3. Page 4 up at the top says AP Biology, Cells and Organelles. So page 4 at the top says AP Biology, Cells and Organelles. That's the page that I want. We're actually going to skip down all the way to the bottom right there where it says eukaryotes. So it's like the bottom quarter of the page. It says eukaryotes, and then right below that, our first organelle is the nucleus. So it's like right here. In bold, all caps, it says eukaryotes. Right there. All right, so we're going to kind of start in the middle of the cell and work our way out. Prokaryotic cells versus eukaryotic cells. What was the thing that eukaryotic cells had that prokaryotic cells don't have? Yeah, a nucleus. Now, there's other stuff that eukaryotic cells have that prokaryotic cells don't, but the reason that we always emphasize the nucleus is because that's what prokaryote translates as. Prokaryote means before nucleus. technically before kernel, but before nucleus. So we think that prokaryotes were around before the nucleus existed. When I was born, the internet didn't exist, so that means when I was like 1 year old or whatever, there was no internet because it didn't exist yet. So you could say I was pro-internet. I was before the internet. Pro means before. If I'm being proactive, if this is glass and it's rolling along the table, I could be like, I wonder what's going to oh. Or I could be proactive and I'd be like, that's going to fall and break. I'm going to get ahead of it before it falls. I'm proactive. All right, so prokaryote means before the nucleus. Eukaryote means true nucleus. It has a nucleus. So prokaryotic cells, like bacteria, they don't have a nucleus. Pretty much everything else does. So if I'm you guys, as long as I know that like prokaryotic cells are bacteria, any other kind of cell that you can think of is pretty much a eukaryote. There are exceptions, but not that you're going to think of. All right, so prokaryotic cells don't need a nucleus. Eukaryotic cells have to have one. Take a moment, talk with somebody around you. Why do eukaryotic cells have to have a nucleus and prokaryotic cells don't need one? What do you think? It's a hard question, unless you've learned it before. All right. Again, I don't think that you would just come up with this on your own. So about the only way that you would know it is if I mentioned it and you were in my honors pre-AP, whatever they called it, when you were in there, class back in the day. Does anybody happen to know why you need a nucleus? What? Why do we need a nucleus? Why do eukaryotic cells need a nucleus? Yeah. True, but like prokaryotic cells have DNA. So DNA doesn't need to be held. A lot of times people will be like, it stores DNA, and you're like, but why don't prokaryotes need to store DNA? Prokaryotes, prokaryotes. Yeah, what do you think? That's true. So maybe the nucleus somehow, because our cells are more complex, maybe they somehow carry out some extra function, something like that. Very logical. It's something different, though. Our cells, you might remember, we do like DNA gets converted into RNA. That was called transcription. Bacteria do that also. RNA is read to make proteins in translation. Bacteria do that also. So their DNA to RNA to protein system works exactly like ours does, except for this whole nucleus part. Here's why. All right. Does anybody know what that is? It's upside down. Yeah. Yeah. So this is a virus. This one happens to be a phage. It's the type that invades bacteria. So bacteria can also get viral infections. This one has color added. If I don't add color under a microscope, looks like that. And in bacteria, the way that they work is they land on the outside of the bacteria. So here's like, a couple dozen that landed on the outside of this bacteria. They squirt in their DNA, and then that DNA starts replicating, and now the cell starts replicating the viruses. Here's again, viruses that infect DNA. You can see this virus landing on this bacterium, and it's like squirting its DNA inside. So, okay, cool. In humans, it's slightly different. Same basic idea, except in us, the whole virus almost always enters the cell. So let me kind of show you what it might look like. Flu looks kind of like this. COVID looks kind of like this. HIV kind of looks like this. So this is like a common picture of like a virus. So let's say I've got a virus, and it's a DNA-based virus. Some viruses have RNA instead. We've never found one with both. So let's say I've got a virus out here with DNA in it. The whole virus usually enters the cell. Okay, that's just for like being technically correct. It doesn't matter. So this is not super important like with polar and non-polar ligands or whatever. So the whole thing enters. I'm going to be a little bit lazy with my drawing and just kind of draw it like that. Then what it does is it kind of like unwraps the DNA. All right. I've also got DNA. So here's my DNA. Like my cell's DNA over here. All right, so it kind of like unwraps its DNA. Cells are really good at reading DNA, and they just do whatever the DNA tells them to do. Just like you, if your DNA says put some melanin in this person's eyes, you're going to have brown eyes, and your cells did that because the DNA told them to. If your instructions for put melanin in your eyes is broken, then you don't, and now you don't have brown eyes. So if there's viral DNA in my cells, then the virus will say like, hey, cell, copy me. And the cell's just like, okay. So now, my cell just starts copying DNA because that's what the DNA told me to do. My cells always do whatever the DNA tells me to do. My computer keyboard, talking to my computer, it's going to show on the screen whatever button I push. If I push the S key, it's going to put an S up there. If I push the T key, it's going to put a T. Whatever I tell it to do, it's going to do. It doesn't ask like, hey, are you sure you want to write this email or whatever? It's just sort of like it's going to do whatever I tell it to do. So now, my cells have this DNA, virus DNA, and then the virus DNA says, make the rest of me, and your cell's like, sure thing, boss. So now, my cell becomes a virus factory. I make 100 viruses, 200 viruses, 1,000 viruses, 5,000 viruses, however many it takes. And then there's so many, my cell explodes. And we call that being sick. So when I'm like, hey, I got a viral infection, why don't I feel good? Well, your cells are exploding. That's why you don't feel very good. Okay, so what's a cell to do? A virus gets inside. It kind of unwraps its DNA. My cells then do whatever the DNA says, which is turn on the virus factory. And now I'm a virus factory. So here is my kind of counteraction. I'm just erasing these for space. Doesn't matter if you erase yours or not or whatever. So now I have an enzyme called DNase. I know it's an enzyme because it ends in ase. Anytime something ends in ase, it's probably an enzyme. If it ends in IN or EN, like we said earlier, it's probably a protein that's not an enzyme. So DNase, you're like, all right, it's either got to construct DNA or destruct DNA. It destroys DNA. Why is it good for me to have DNase in my cell then? Yeah, stop this. So let's say this virus gets inside and it starts trying to copy its DNA. What do you think happens if it bumps into DNase? What happens to the DNA? Yeah, it gets like destroyed. Great. So is that a good thing for my cell? That the viral DNA gets destroyed? That's a good thing for my cell. Not for the virus, but for my cell. However, is there a problem? Don't I have DNA? Won't DNase destroy my DNA? Yes, it will. So how do I avoid that? The nucleus. The function of the nucleus is to protect DNA from DNase. DNA does not need to be held. Bacteria have DNA and it works fine without being held. So the function of the nucleus is not to hold DNA, it's to protect DNA from DNase. That's the function of the nucleus. Now, as we go further along in the year, we'll end up learning a few other functions too, but for right now, the function of the nucleus is to protect DNA. Protects DNA. So now, if my DNA ever leaves the nucleus, isn't it likely to get wrecked by DNase? Yeah, so if my DNA ever leaves the nucleus, I have to first dismantle DNase, but then I'm susceptible to viral infection, so that's risky. Okay, here's the problem. I've got to build a protein. And let's say this DNA right here, here's a gene that codes for something that I need. It codes for insulin. Insulin protein. If you can't quite read that from where you're at, it just says insulin. I just kind of pretend that this chunk of DNA is the insulin gene. All right, so I need to build insulin protein. But for insulin protein to work, insulin protein has to like leave the cell and I have to dump it into my bloodstream. Let's say that insulin is this big. So here's the code for insulin. I need to build insulin protein. Does it look like stuff might be able to get in and out of the nucleus? Based off of the way that I drew that, does it look like there might be ways for things to get in and out of the nucleus? Yeah. So I've got these little nuclear pores. The original NPC, the nuclear pore complex. So people always be like, "You're such an NPC," and I'd be like, "A nuclear? Did that kid just call that kid a nuclear pore complex?" So we've got these kind of nuclear pores. We can let stuff in and out. Cool. Here's insulin protein. For it to do its job, it has to like get out of the cell. If I read the insulin gene and I build the protein in the nucleus, does it look like it can get out of the nucleus if it's this size? No. So here's the problem. Most proteins are too big to exit the nucleus. But the code for that protein is in DNA. The code for everything's in DNA. So I have to somehow find a way to like get the DNA code out here because I have to build the protein out here, but DNA can't leave. So what do? I've got to get the insulin instructions, but they have to be out here because I have to build insulin out here. It's so big, insulin is like stuck in the nucleus. But if I build insulin out here, everything is cool. Yeah. It is. That's why RNA exists. You're exactly right. So what I'll do is I'll make an RNA version of this insulin gene. RNA can safely leave the nucleus. Now, I can read RNA and I can build the protein and I'm good to go. So that's why RNA exists. DNA has the instructions I want, but I have to build out here in the cytosol. But if DNA leaves, DNA wrecks it. So DNA has to be in here, but I have to read the instructions out here. So I just convert them into RNA language. That's why RNA exists. Before that, it seems kind of dumb. They're like, all right, here's DNA. The first thing we do before we read it is we convert it into RNA, and then we read that. And you're like, why not just read the DNA? Well, we can't. So RNA leaves, and we can build the protein. So why do we need the nucleus again? Protects DNA from DNase. But doesn't the genetic message need to leave the nucleus? Yeah. DNA can't leave the nucleus. RNA can. Does DNase destroy RNA? No. Because if it did, what would it be called? What would its name be? RNase. So it totally makes sense. All right. I'll ask you one last question, and then we'll pack up. Who was it that read RNA? It's the name of an organelle. Who was it that read the RNA and built protein from it? Do you remember? Yeah. Ribosomes. Yeah. So we'll pick up next time with ribosomes. Ribosomes, to me, they kind of look like hamburger buns because it's got like a big subunit up top and a small one on the bottom. And ribosomes are going to read that RNA and build the protein that it says to build. Now, if you're looking at your notes on nucleus and you're like, hey, there's a bunch of stuff that he skipped, true. I'm going to kind of give you a broad overview of all these organelles working together, and then we'll come back and like hit the more specific details on each one. So that's it for now. See you guys later.